Hello my dear friends, welcome to Live Prep. In the previous lecture, we studied about what are the various factors that are affecting the exchange of gases. So there we studied that, so partial pressure gradient is one important factor, and then solubility of the gases in the water, that is also one important factor, and finally we studied about the thickness of the diffusion membrane. So these are the various factors that are affecting the transport of gases or the diffusion of gases, not transport, diffusion of the gases across the membranes. So that's what we have studied in the previous lecture. Now in this lecture, we are studying mainly on the transport of the oxygen. How exactly oxygen is transported. So here we are focusing on transport of oxygen and transport of carbon dioxide. But in this lecture, we will focus mainly on transport of oxygen. But here also I am dividing this into two parts. So here in one lecture, we will study some basic information. Yes or no? So in the next lecture, we will go with that is also regarding transport of oxygen only, but there in this lecture we will study basic information regarding the oxygen dissociation curve. But in the next lecture, I will tell you what are the factors that are affecting and some other important points. Clear? Because in the single lecture if you are doing, so it will become a little bit lengthier. So that's why I am planning to divide it like this. Anyhow, for transport of carbon dioxide, one lecture is enough. So there we will study about this particular information. Clear? And now let us see overall. So in our body, so how exactly these gases called oxygen and carbon dioxide are transported. So for the transport of oxygen, if you see, majority of the oxygen, that means 97% of oxygen is transported by the RBC. Inside the RBC, hemoglobin is there. So that oxygen will bind with hemoglobin and it will form oxyhemoglobin. So in this form, 97% oxygen is transported, whereas the remaining 3% is directly solubilized in that blood plasma. In the plasma, it will be solubilized. Already in the previous lecture, we studied, no, there is some solubility, some gases are solubilized. So in the solubilized form, 3% of oxygen will be transported. But please remember, this solubility of oxygen in the water is a physical process. And please remember, here we are not talking about any enzyme. Please remember this. Next one, transport of carbon dioxide. So carbon dioxide is transported in three important mechanisms. One is majority of the CO2 is transported as bicarbonate. Anyhow, mechanism separately we will study. Simply the values you please remember. 70% of CO2 is transported as bicarbonate, whereas 20 to 25% CO2 is transported as carbaminohemoglobin. That means attached to hemoglobin through the RBC, it will be transported to the lungs. And then whereas 7% of the CO2 is dissolved in the blood plasma. So in this way, so it will be transported, CO2 is transported from the tissues to the lungs, whereas oxygen is transported from the lungs to the tissues. This is what. And now we are focusing about this particular oxygen transfer. Anyhow, dissolved form means for that we are not studying anything, simply it will be dissolved. Okay. So here we are studying, before going with the individual explanation, first we will see the general information given in NCERT. Blood is the medium of transport for oxygen and CO2. So blood is the major one that is carrying these particular gases to various locations. About 97% of oxygen is transported by the RBCs in the blood. How it is transported? Within the RBC, hemoglobin is there. Oxygen will attach to hemoglobin and it will form oxyhemoglobin. That means in the form of oxyhemoglobin, this 97% oxygen will be delivered. Next one means it will be transported. The remaining 3% of oxygen is carried as a dissolved state through the blood plasma. So in the blood plasma, it will be dissolved and it will be transported. This is what about oxygen. For CO2, nearly 20 to 25% of CO2 is transported by the RBCs. How it is transported by RBCs? As carbaminohemoglobin. Slowly we will study all this. Whereas 70% of this CO2 is carried as bicarbonate, and about 7% of CO2 is carried in the dissolved state through the plasma. So these are the various important percentages they give. So you need to compulsorily remember these values. This is compulsory. And now we are specifically focusing on transport of the oxygen. And in this lecture, we will focus mainly on oxygen transport with the help of this hemoglobin. So basic structure of hemoglobin we will study, oxygen dissociation curve. Up to that we will study. Remaining points of the oxygen dissociation curve we will see in the next lecture. Clear? First we will see dissolution of the gases. Already I told you in the previous lecture also, solubility is one important factor. CO2 has more solubility when compared with oxygen. That point also we studied, yes or no? And even for a particular gas also, whether it is oxygen or CO2, whatever it is, even for a particular gas also, If it is having high partial pressure or high pressure for that particular gas, then more amount of gas will be solubilized. If the pressure is very less amount of gas will be solubilized. That means if more pressure is there, more solubility will be there. If less pressure is there, less solubility will be present. But anyhow, whatever the soluble pressure that is present for the oxygen, for that approximately 3% of oxygen is solubilized within the blood plasma. This is how it will be solubilized. It will be solubilized and it will be transported, okay, to the various tissues. That's it. With this, transport of oxygen through the blood plasma is completed. We are not worrying much about this. But the major point is transport of oxygen through the RBC. Inside the RBC, what is present? Hemoglobin is present. Okay. Calculation things and all the things separately we will study. So don't worry. Basic information we will study. For the hemoglobin, we need to study some important points. What are the points that we need to study? What are the chains that are present within the hemoglobin? I will tell you this information point by point. So please try to understand this. So generally we are studying about the hemoglobin here. Hemoglobin, and this hemoglobin has 4 chains. Hemoglobin has 4 chains. Out of these 4 chains, please remember, in the biomolecules, when we studied about the protein primary, secondary, tertiary, quaternary structure, there we used this name. Hemoglobin has 4 chains. Out of these 4 chains, 2 chains are of same type and 2 chains are of different type. And what are these two? 2 are alpha chains. 2 are alpha chains, whereas 2 are beta chains. 2 are alpha chains and 2 are beta chains. Please remember this. And now in this way, generally for the hemoglobin, whenever we are writing, we write this hemoglobin as alpha 2, beta 2. So in this way we are writing, that means 2 protein chains. Already when we studied about biomolecules, there we studied about this. So 2 alpha chains. For each alpha chain, if you see, for one alpha chain, it is having tertiary structure. Other alpha chain, it is having tertiary structure. And the beta chains, individual beta chain, each beta chain will have tertiary structure likewise. So this is alpha, alpha, beta, beta, like this. So individual protein, if you see, it is having tertiary structure, but when they are interacted, here, ultimately, like this I am writing. So now this entire structure is called as quaternary structure. This is what the information already we studied in the biomolecules. So in this way, within the hemoglobin, total 4 protein chains. These chains are nothing but the proteins. These are the protein chains. Okay, they are the proteins. Out of these 4, 2 are same type. They are called as alpha proteins or alpha chains, and 2 are of same type. They are called beta chains or beta proteins. This is how they are present. Normal hemoglobin means this is the hemoglobin that we are studying. It is having 2 alpha and 2 beta chains. Clear? This is the one important point that you need to understand. Next important point. What are the types of hemoglobin here? Why we are focusing like this? Because we will study about oxygen dissociation curve. So here I will add another type. So that's why observe carefully here. Total 3 types of hemoglobin we are studying. I am writing HB, capital H, small b, that means hemoglobin. Okay, I am not writing full. This is in short I am writing. 3 types of hemoglobin we are studying. What are these? Adult type, and then fetal type. Fetal type, you need to understand the concepts behind these also. You need to understand this. Okay, that's why I am breaking this particular transport of oxygen into 2 lectures. You need to understand the logic. Okay, adult type. What is adult type and what is fetal type? You need to understand this. I am giving this information, and this information we will use in so many other chapters also. Please understand this. Okay, so please observe carefully. Suppose mother, okay, mother, okay, so pregnant mother. So inside this pregnant mother, we are talking about the development of a fetus. This is the fetus. Fetus is growing here. Understood or not? Now here, between the mother and the fetus, there is a connection, and this connection is called as placenta. Placenta, yes or no? And this placenta and this fetus, they are connected by a long tube-like structure that is called as umbilical cord. But anyhow, I am not writing. So this is what the point is. Now this is the fetus. Now please observe carefully. When this fetus is slowly developing, various organs will be developed. Please understand this concept. This concept once again linked with the next chapter, that is called body fluids and circulation. There once again I will tell the same thing, so please listen very carefully. So fetus is developing inside the mother's womb. Yes or no? Now fetus is developing. So when it is keep on developing, various organs are slowly they are developing. Very good, nice. When they are developing, even within the fetus also lungs are developed. Even within the fetus also lungs are developed. Even though lungs are developed, they are not functional. Please understand this particular logical concept. So based on this, in body fluids and circulation, for the heart we will study some extra connections. So please understand this. Even though lungs are developed here, they are not working. Now you just imagine here, mother is having lungs separately, and mother's lungs are working, so that breathing process is taking place. So mother is getting the oxygen and releasing the CO2. Nice. But how this fetus will get oxygen and how this fetus will remove this CO2? Observe carefully. So when it is present inside the mother, are these lungs functional? No, they are not functional. Now the question here is how this fetus will get oxygen from oxygen. This is what the question is. Observe carefully. Now you just listen very carefully. Now within the mother, for the mother blood circulation is there. So mother's blood circulation will come up to the placenta. Mother's blood circulation is coming up to the placenta. Within this mother's blood, there are some RBC. Inside this RBC, hemoglobin is present. The same concept once again useful in this lecture also at the end. So please listen very carefully. So within the mother, within the blood, RBCs are present. Within the RBC, hemoglobin is present. Nice. Now for the fetus also, inside the fetus also, RBC are present. And this inside this RBC, there is a hemoglobin. Understood or not? Now the question here is, are these two hemoglobin, that means hemoglobin of mother and hemoglobin of the fetus, are they same? Hemoglobin of mother and hemoglobin of fetus, are they exactly same? Anyhow, this mother is having 2 alpha chains, 2 beta chains, correct? But here what about the fetus? It is not like this. Fetus is having 2 alpha chains, correct? But not 2 beta chains. Here fetus is having 2 gamma chains. Very important, very important. At least understand the logic. So mother is having alpha to beta 2 type of hemoglobin, but the fetus is having alpha to gamma 2 type of hemoglobin. So this type of hemoglobin present in the fetus is called as HB. HB means hemoglobin. F means fetal. It is called as a fetal hemoglobin. The hemoglobin present in the fetus is called fetal hemoglobin. But mother means adult, right? So the hemoglobin present in the mother is called as HbA. A means adult. A means adult. You need to remember A means adult, because this particular content once again we will study in class 12th chapter, that is called as principles of inheritance and variation. In principles of inheritance and variation, we will study about sickle cell anemia condition. There we will study about HbA, HbS, like that we will study. So A means adult. A means adult. Please remember this. A means adult. So this is the adult type of hemoglobin and this is the fetal type of hemoglobin. Understood or not? So in this way, 2 different types are present. Now the question here is, why fetus is having a different type of hemoglobin? What is the logic here? Now observe carefully here. Even though lungs are present within the fetus, these are not working. Lungs are not working. Then if lungs are not working, how fetus will get the oxygen? Fetus should get oxygen from the mother. Mother is getting through mother's lungs. Yes or no? Through the lungs, mother is getting the oxygen, fine. And now it is going in the blood supply. And that blood supply is coming up to the placenta. And now what fetus will do? Fetus will get oxygen from the mother's blood. So in the mother's blood, alpha to beta 2 type of hemoglobin is present, and to this hemoglobin, oxygen will be attached. Now it is coming up to this. And now this fetal hemoglobin is having alpha to gamma 2. And now you please understand this. Already here oxygen is attached to this one. But fetal hemoglobin should be more powerful. That means fetal hemoglobin should get oxygen from mother's hemoglobin. Understood or not? That means from here it should get. That means here the affinity, the binding capacity, affinity of oxygen to bind with fetal hemoglobin is more. That means it can easily dissociate from adult hemoglobin and it will readily bind with fetal hemoglobin. Then only fetus can get oxygen from the mother, or else it cannot get. That's why a different type of hemoglobin is present in the fetal RBC. I hope now you understood the logic here. Why fetus is having different type? Because this hemoglobin should be having more binding capacity. Then only it can take oxygen from the mother. That is the reason why a different type of protein, that is called gamma protein, is produced. That's why alpha to gamma 2, that is called fetal hemoglobin, is present in the fetus. Please remember this one. In this lecture itself we will study about oxygen dissociation curve. There I will connect this point once again. So now between these two, he is having more binding affinity. Fetal hemoglobin has more binding affinity for oxygen. That means oxygen will quickly bind with fetal hemoglobin when compared with adult hemoglobin. Clear? This is what the logic is. So in this way, different types of hemoglobins are present. So one is adult, other one is the fetal. So now we will go back to this. So fetal type, it is called as HbF. So what are present in this? Two alpha chains and two gamma chains. So alpha to gamma 2. This is the fetal type. Now in the adult type again, two types are present. Adult type again, two types are there. HbA1 adult 1 type. Other one is called HbA2 adult 2 type. In majority means what? 97%, approximately 97% will be adult 1 type only. And that is what regularly we are studying. This is alpha to beta 2 type. The regularly we are studying type is the alpha to beta 2, that is simply called as adult type. More specifically, it is called adult 1 type because more present. Other type is very less, just 3%, and it is alpha to delta 2. Alpha to delta 2, a different type of protein is present. So in this way, different types of hemoglobin molecules are present. Adult 1 type is having alpha to beta 2, adult 2 type is having alpha to delta 2, and fetal type is having alpha to gamma 2. Out of these three, what is the compulsory information? Adult 1 type, alpha to beta 2 is compulsory. Now we are studying information for this particular one only. And regarding adult 2 type, even though you are ignoring, no problem, you can ignore this. But for a fetal type, even though not mentioned in NCERT, significance you need to understand. In the oxygen dissociation curve, I will tell you the significance. So on which side you are getting this, what happens to P50 value, all the things with respect to these two you need to compare. So that's why at least some basic information you please remember for fetal type. Though not mentioned, I am suggesting that please remember the information regarding this. In this way, three important types of hemoglobin molecules we are studying. So this is about the types of hemoglobin. Clear? Next one. So anyhow, since we are talking only about the adult 1 type, alpha to beta 2 type, what are the genes that are responsible here? Because in the class 12th chapter, principles of inheritance variation, they mentioned it. So in simple, I will try to tell you that. And along with this, there itself you will study about sickle cell anemia and thalassemia. What are these issues? Because they are also related with hemoglobin. So please understand this particular concept. In simple, I will tell you. Anyhow, there we will study more details. So what is this particular one? Alpha chain, beta chain, they are proteins. They are proteins means they are produced from some mRNA, and this mRNA is produced from DNA and it is called as gene. Yes or no? That means genes are producing these proteins. So alpha chain is produced from a specific gene, beta chain is produced from another gene. So in this way, genes are there. What are these genes? Suppose you are talking about the human, we are having total 46 chromosomes. So here we are focusing on chromosome number 11. Two are present because pair are present, right? So pair of chromosomes are present because one is obtained from father, other one is obtained from the mother. So in this way, pair of chromosomes are present. On the chromosome number 11, number was also given in NCERT, so you should remember this, okay? And in the same way, we are studying another one, that is the chromosome number 16. Chromosome number 16. Now here observe carefully. Beta chain gene is present on chromosome number 11. Alpha chain genes are present on chromosome number 16. Observe carefully. Now here, for the beta chain, there is one gene. One gene means two alleles are present. This is called as HBB. HB, hemoglobin B, B for beta, beta chain. So the gene name is HBB and it is present on chromosome number 11. One gene is present but it is having two copies, one copy on paternal chromosome, one copy on maternal chromosome. That's why these two are called alleles. So for beta chain, one gene, that means two alleles are present. Two alleles are present. Remember this information at least. Anyhow, in the genetics we will study here. But for the alpha chain, the concept is here. This is one gene. This is called as HbA1. A for alpha. Hemoglobin alpha 1 gene. Another one is HbA2 alpha 2. Now here what happens? So for the alpha chain gene, the alpha chains, two genes are present. For this gene, two alleles. For this gene, two alleles. So for two genes overall, how many alleles? Four alleles are present. Four alleles are present. Remember this information. Very, 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 very important for examination. Okay. This is what the information they said. And these alleles they will produce alpha chain, sorry, beta chain. And these four alleles they will start producing alpha chains. So in this way, in the case of adult individual, alpha chains are produced and beta chains are produced. So in this way, two alpha chains, two beta chains combinedly they are producing this hemoglobin. So in this way, this is at the gene level concept at the gene level. And now I have told you two more names for the diseases. What are these two names? One is sickle cell anemia, one is thalassemia. First I will tell you thalassemia. What happens in thalassemia? These are the genes. Suppose there is a mutation in the gene. Because of the mutation, beta chain is not produced. Understand this. Due to some mutation, beta chain is not produced. If it is not produced, that is called beta thalassemia. That means hemoglobin content will decrease. If beta chain is not there, what happens? The hemoglobin level will decrease. So that type of problem is called as beta thalassemia. Second problem. This is also thalassemia. Because of the mutation in these genes, if alpha chain proteins are not produced or their number is decreased, alpha chain not produced or alpha chain decreased, that is called alpha thalassemia. Here due to mutation, beta chain is not produced, beta chain number is decreased. Whatever it is, overall there is a decrease. That is called as thalassemia. So in the thalassemia what happens? Less production or no production, less or no production of either alpha chain or beta chain. That is the alpha and beta thalassemia. So in the thalassemia, hemoglobin protein chains, alpha chain, beta chain, production is decreased or production is not happening. That's why it is called thalassemia. Okay, fine. Then what is sickle cell anemia? In the sickle cell anemia, concept is related only with beta chain, not with alpha chain. Sickle cell anemia concept is applicable only for beta chain. Here what happens? Here the type of mutation is a different type of mutation. The problem here is protein is produced. Even though there is a mutation, still protein is produced. But within that protein, amino acid is changed. Even though protein is produced, amino acid is changed. So that's why that will be coming under sickle cell anemia. So that's why in the case of thalassemia, quantity, quantity is decreased. That's why thalassemia is a quantity problem, quantitative problem. In thalassemia, no problem with quantity. Number of proteins are normal, but the protein structure is affected due to amino acid difference. That means quality of protein is affected. That's why sickle cell anemia is qualitative problem. Thalassemia is quantity, number wise problem. Number of proteins are decreased. Sickle cell anemia, there is no problem with number, but there is a problem with the quality of the protein because amino acid is changed. So that's why this is sickle cell anemia. So in this way, with respect to genes, with respect to these diseases, whatever I said now, same information with more and complete details we will study under the genetics. So in this way, this hemoglobin content is very, very important for the next coming lectures also. You need to compulsorily remember this. So this is what we are talking about, the hemoglobin. And now we will go with the normal structure of this one. In the hemoglobin, generally we are talking about two alpha chains, two beta chains only. But apart from this, there is one protoporphyrin 9 ring. So here, if you are observing this, so in the hemoglobin, protoporphyrin protoporphyrin 9 is present. If you can remember this name, try to remember this. If you feel that this name is difficult, ignore it. But my suggestion is better remember this, because this is one of the important names with respect to hemoglobin. Within the hemoglobin, anyhow, globin chains we studied, alpha chains, beta chains. But that heme is present, right? So now it is actually protoporphyrin ring. In this protoporphyrin ring, at the center, iron is present. And this iron, on four sides, it is connected with some pyrrole rings. Pyrrole rings are present. So in this way, in this particular hemoglobin, iron center is present, and that is connected with the four pyrrole rings. And in this way, it is ultimately related to protoporphyrin 9 ring, and this ring structure is not needed. At least remember that it is having iron center, iron center. This is first important point. Second important point, the oxidation state of iron is Fe plus 2. Oxidation state of iron is Fe plus 2. That means it is a ferrous form. If it is Fe plus 3, that is ferric form. But here in the hemoglobin, it is present as Fe plus 2, ferrous form. Very, very important. Please remember this. This is how it is present. And now again these are connected with that globin chains, alpha chains, beta chains. So in this way, in the hemoglobin, both protein part, non-protein part is there. Protein part are that alpha chains, beta chains. Please remember, this is not enzyme, so don't use apo enzyme cofactor terms, that is not applicable. So this is not enzyme, okay. So in the hemoglobin, protein chains are present, and a non-protein part, that is called as protoporphyrin 9 ring, having the heme center is present. This is how it is present. Is it clear? So this is how we are talking about the hemoglobin structure. This is the basic structure of hemoglobin. This is how it is present. And now we are talking about the oxyhemoglobin. When oxygen is binding with this, particular hemoglobin, then where exactly oxygen is attached? This is another important point. Many students are not focusing on this. They are thinking that yes, oxygen will bind to hemoglobin, but where exactly? Observe this. This is the Fe plus 2, right? To this Fe plus 2, now here oxygen is attached like this. O2 is attached directly to the iron Fe plus 2 that is present within this particular ring. Understood or not? Now here another point is, one particular heme group is binding with one oxygen molecule. One oxygen molecule. These names are very, very important. Okay. One oxygen molecule means O2. This is one oxygen molecule. One oxygen molecule has how many atoms? That is equal to 2 oxygen atoms. You need to remember these words very carefully. Okay. Very, very important. Observe carefully. One molecule of oxygen has 2 atoms of oxygen. Please remember here. So in this way, in one hemoglobin is actually a tetramer. 4 are present together. 4 are present together. If 4 are present together, so one is binding with one oxygen, second one will bind with another oxygen, third one will bind with another oxygen, fourth one with fourth oxygen. So in this way, one hemoglobin binds how many? How many of these? Binds 4 oxygen molecules. Please write these sentences very carefully. Since it is present as a tetramer, 4 units, each one is binding with one oxygen. So that's why one hemoglobin, overall it is binding with 4 oxygen molecules. That means it binds with 8 oxygen atoms. 8 oxygen atoms. Understood or not? That is the reason why generally how we will write HbO2 4 times we are writing. It is the O2 4 times we are writing. HbO2 4 times. So hemoglobin is binding with total 4 oxygen molecules. That means 8 oxygen atoms. So in the examination, whether they are asking question for molecules or atoms, you need to clearly observe the question carefully. Please don't argue with us later on. So one molecule of hemoglobin will bind with 4 oxygen molecules or 8 oxygen atoms, you are saying. So that's why generally we are writing like this, HbO2 4 times. Even though generally you are writing HbO2, technically it is 4 times, because 4 molecules of oxygen are attached to one hemoglobin molecule. This is one important point. So in this way, oxygen is attached to hemoglobin, so that now it is forming oxyhemoglobin. Oxygen attached to hemoglobin. That's why it is oxyhemoglobin. This is what. And now here I am telling another point, that is regarding the spelling. Some students have the habit of writing oxyhemoglobin, HAEMOGLOBIN, whereas some students have the habit of writing oxyhemoglobin. How you are writing hemoglobin? Directly you are writing HEMOGLOBIN, hemoglobin, and here you are not writing HAE, you are writing HEMO. Is there any difference here? Is there any difference here? I told you in the initial introductory lectures, okay? So before starting the chapters, I told you some introductory lecture, there I told about this. Please remember, both the spellings are same, there is no difference. In the case of British English, we are using this HAEMO. In the case of US English, we are using HEMO. Both of them are exactly same. Some students are thinking that both of them are different. They are not different, both of them are exactly same. Okay, same way here, fetus, FOETS, FETUS, both of them are same only. Yes or no? So generally in the US, we are writing like this. In the British, we are writing like this. Meanings are same only. In the spellings, there are some small difference. Same way here also. HEMO you can write, HAEMO also you can write. But in the NCERT, I am telling you, NCERT is following British. Okay, so that's why they are giving these spellings. But when we are preparing some question papers, generally we are installing in the US. When we are installing that English, so we are installing the US format. So that's why even though we are typing like this, it will show the red, so that we will, when we are going for auto correction, it will show like this. Both of them are same only. Don't get confused. Please understand these particular things. Okay, in this way, oxyhemoglobin we are writing. Now I am having one particular very important question. Many students are not focusing on this. Even teachers are also not saying this. But what is that particular question? Oxygen is attached to hemoglobin, fine. And the molecule is called as oxyhemoglobin, fine. But please remember, generally in the chemistry we are studying this. Addition of oxygen is called as oxidation. Removal of hydrogen is called oxidation. Removal of electrons is called as oxidation. This is what you are studying. Yes or no? This is what you are studying, right? Yes. Now my question is, here oxygen is attached to this hemoglobin and it is forming oxyhemoglobin, fine. But my question is, attachment of oxygen to hemoglobin is oxidation process or oxygenation process? Question is very clear. Question is very clear. You need to understand this because it is concept based. Please understand this. Attachment of oxygen to hemoglobin will result in oxyhemoglobin, correct? But this process of attachment, during this process, what happens? Oxidation occurs or oxygenation occurs? This is very important point. Many students are not focusing on this. It is very important for competitive examination, so observe carefully. When oxygen is not attached, now let us see. When oxygen is not attached, that means it is simply hemoglobin. It is hemoglobin. When oxygen is not attached, it is hemoglobin. Within the hemoglobin, what is the oxidation state of iron? It is Fe+2. When oxygen is not attached, then oxidation state of iron is Fe+2. Please remember this. But when it is forming oxyhemoglobin, that means when oxygen is attached, question here is whether it is oxidation or not. How you will know this? If iron is oxidized, oxidized means what? It is having two important oxidation states, Fe+2 to Fe+3. Yes or no? This is the ferrous form, this is the ferric form. Yes or no? So if Fe+2 is converted to, if I am using a word called if Fe+2 is converted to Fe+3, you can say that it is oxidized. It is oxidized. Then you can say that it is oxidation process. But what is the answer here? In the oxyhemoglobin also, iron is present in Fe+2 only. Please understand this. When oxygen is not attached, then it is anyhow present in Fe+2. Even though oxygen is attached, still it is present as Fe+2 only. That means oxidation state of iron is not changed. It is not changed. It is not converted into Fe+3. Still it is present as Fe plus 2 only. So can you call it as oxidation process? No. It is not oxidation process. Simply it is the attachment of oxygen without causing any change in oxidation state. That's why it is called as oxygenation process. Please understand these names. It is not oxidation, it is oxygenation. Simply oxygen is attached without causing any change in the oxidation state. So that's why oxyhemoglobin formation is oxygenation process, not oxidation process. Very, very important thing. Please understand this. This is one important point that you need to understand. And where exactly this particular step is happening, oxyhemoglobin formation. This oxyhemoglobin formation is taking place at the blood vessels of alveoli. At the blood vessels of alveoli. In the alveoli, oxygen is more. It is having 104 mm mercury. Yes or no? Now let us see this. This is the deoxygenated blood. Deoxygenated blood. In the deoxygenated blood, partial pressure of oxygen is 40 mm Hg, 40 mm mercury. Yes or no? This is what. Now here what happens from high pressure to the low pressure. Now oxygen is keep on coming into this one. It is keep on coming, so that now the partial pressure is raised to, sorry, PO2 is raised to 95 mm mercury. In this way, here less oxygen is there, here more oxygen is present. Now that's why it is oxygenated. Oxygen is added to this one, so that's oxygenated blood. Okay, at the tissues oxygen is used up. So once it is used up, D means what? D means what? D means removal. Oxygen is removed, not completely removed, some amount is removed. Where it is going? It is going to the tissues, so that now again it is converted into deoxygenated blood. Now this is at the tissues. At the tissues will use oxygen. Yes or no? So once oxygen is removed from this, now it is called deoxygenated. But please remember, from the blood you are talking oxygenated, deoxygenated blood. But our main focus is with respect to hemoglobin. Yes or no? So here we are focusing about the hemoglobin. So we are focusing on hemoglobin. So hemoglobin, to this hemoglobin if oxygen is attached, oxyhemoglobin. When oxygen is removed from this, then actually CO2 will attach. CO2 will attach with hemoglobin in form carbaminohemoglobin. There also it is Fe plus 2. When CO2 is attached to hemoglobin, then also it is Fe plus 2 only. So iron oxidation state is not changed. When it will be changed, under the extra information, at the end of the lecture I will make one separate lecture for extra information. Some points that were not mentioned in NCERT but may be useful, that content I will make one separate lecture. So in that lecture I will discuss about that particular point. So whether it is oxyhemoglobin or carbamino, CO2 attached to hemoglobin, carbaminohemoglobin or normal hemoglobin, whatever it is always present in Fe plus 2 only. So that's why attachment of oxygen to iron is oxygenation process, not oxidation process. Wordings are very, very important. Observe very carefully. Clear? This is what about the hemoglobin. And now to this hemoglobin, oxygen should be attached. And for that, we are talking about one oxygen dissociation curve. We are talking about oxygen dissociation curve. Please remember this particular curve. This is a graph paper. On the graph paper, we are making these dots and then we are making this particular curve. So it is an experiment. We are doing this particular experiment in the laboratory. Please understand this. This particular oxygen dissociation curve, whatever you are saying, this oxygen dissociation curve, we are actually performing this particular one in the laboratory. In the lab we are doing this particular experiment. So in this experiment, what actually we are doing, on the x-axis we are maintaining partial pressure of oxygen. This is low and this is high, low to high. Understood or not? On the y-axis what we are mentioning, on the y-axis we are mentioning percent saturation of hemoglobin with oxygen. When oxygen is binding with hemoglobin, after some time it will be saturated. So how much it is saturated? What is the percentage saturation of hemoglobin with oxygen? Here you are providing oxygen, you are taking a particular quantity of hemoglobin and you are keep on giving the oxygen. Once you are giving a particular partial pressure of oxygen, then how much it is saturated? When you are increasing the oxygen supply, then how much it is saturated? So in this you are keep on increasing, then when it will be fully saturated. Likewise, we want to know what is this binding ability, what type of relationship it is showing. Will you get a straight line like this, or will you get a different line, and why you are getting a different one? These are the points we need to discuss. So when experimentally when you are performing this is what the graph they got it. So here this is 10, 20, 30, 40, 50, 60, 70, 80, 90, 20, 90 and then 100. In this way, this is the partial pressure of oxygen that we are giving. This is the partial pressure of oxygen that we are providing in that experimental condition. Now, what the scientists have observed, when it is 10 mmHg approximately, as per this particular diagram I am giving you, I am not giving the exact values with the dot points and all, simply rough values we are getting it from the diagram. When it is 10 mmHg, how much it is saturated? 10%. On the x-axis it is percent saturation rate, 10% hemoglobin is saturated. Next one, when it is 20 mmHg, it is approximately 30% saturated. See here, there is a sudden increase here. And when it is 30 per mmHg, you can see that it is approximately around 55 it is coming, right? It is not exactly 60, around 55 you are getting this. Yes or no? So or 58, somewhere 58, because this is 50 and it is more than 55. So you can think that it is approximately 58% saturated. That means sudden rise is there. When it is 40 mmHg, you can see this. This is 60, this is 70. So this is middle of this one. So you can say 75% approximately saturated. So when it is 40 mmHg, it is approximately 75% saturated. When it is 50 mmHg, you just see this. Around it is around, suppose some 83 somewhere, 83, 84 somewhere you are getting based on this. Yes or no? Next one, when it is 60 mmHg, you are getting approximately 90% saturated. See here, at 60 mmHg itself, 90% saturated, and after that it is becoming little bit slow. 70%, 70 mmHg, so around it is 9, so this is 90 and this is around 94 somewhere you are getting it. When it is 80 mmHg, you are getting around somewhere 96, 97 you are getting. When it is 90 mmHg, around 98, 99% you are getting. When it is 100 mmHg, you are getting almost 99 to 100% saturation. This is what the graph. So in this way, scientists in the experimental conditions, they have provided this particular partial pressure of oxygen, and then in that particular partial pressure, what is the percentage saturation, how much, what is hemoglobin should be saturated with the oxygen binding. So how much in the percentage they have given. So this is what the percentage saturation we are getting. So that's why in this way if you are observing, so here we are getting a curve like this. We are getting a curve like this, and this type of curve is called as sigmoidal curve. This type of curve is called as sigmoidal curve. So that's why oxygen dissociation curve. Oxygen dissociation curve. Oxygen dissociation curve is having sigmoidal shape. This is what the shape that we are getting here. In this way, the sigmoidal shape. So initially, though it is slow, all of a sudden it is rising, again it is becoming decreased. So in this way, it is going like this, so that now it is fully saturated. That's it. So in this way, they made a graph, and this graph is called oxygen dissociation curve. But please remember, when you are talking, when you are going from left to right, it should be called as association, because oxygen is keep on binding to hemoglobin. But please remember, technically we are focusing from right side to left side. Right side to left side. Here it is more saturated, means more oxygen is binding with hemoglobin. But when you are moving from right to left, when partial pressure is decreasing, now what happens? Saturation of hemoglobin is decreasing. What is the meaning of this? Saturation of hemoglobin is decreasing means hemoglobin is releasing the oxygen. That's what the meaning, right? Suppose so and so amount of hemoglobin is there. When more and more oxygen is given, left side to right side you just see, left side to right side you just see. More and more oxygen is given, it is going and binding to hemoglobin. Now more oxygen, hemoglobin is saturated with oxygen. Reverse. If the partial pressure of this is what important for examination. Observe carefully. When partial pressure of oxygen is keep on decreasing, what is happening to this value? Keep on decreasing. That means if partial pressure of oxygen is decreasing, oxygen is dissociated, separated from the hemoglobin, so that it is donating the, or it is releasing the oxygen. Yes or no? This is what they are saying. It is releasing the oxygen. So hemoglobin, in simple I am writing, I am not worrying about O4, O2 4 times, okay. Now here what happens? Hemoglobin and oxygen are separated. Now the separated oxygen will diffuse into tissues, will diffuse into the tissues. This is what the point they are saying here. So in this way, oxygen at the tissues, it is separated from the hemoglobin and then it is going into the tissues. Understood or not? This is what the important point that you need to notice here. And now this is the experimental condition. Though it is experimental condition, what are the partial pressures of oxygen that we already studied? What are the partial pressures of oxygen that we already studied? Observe carefully here. This point once again in the next lecture also I will tell you, but please observe carefully here. This is the alveoli. This is the blood circulation. Okay, and it is going to the heart, and again from the heart it is going to the tissues. Okay, and again from the tissue deoxygenated blood is coming to the heart, and again from the heart it is going to the lungs. Clear? Now let us see this. This is oxygenated blood. This is deoxygenated blood. Anyhow, in the next lecture I will tell you calculations. At the time it will be useful for you. So here I will tell you the simple point. So here in the alveoli this is the alveoli. Inside the alveoli blood is not there, right? So around the alveoli blood circulation is present. Now here within the blood hemoglobin is there. So we are focusing only on blood. Blood means what? Oxygenated and deoxygenated blood. What is the partial pressure of oxygen in the case of this oxygenated blood? It is 95 mm mercury. It is 95 mm mercury. But what is the partial pressure of oxygen in the deoxygenated blood? It is 40 mm mercury. So even though this graph is an experimental graph, we are worrying about two important pressures. We are worrying about two important partial pressures. What are these two important partial pressures? One is 95 mm mercury related to oxygenated blood. That means within the oxygenated blood, this is the oxygenated blood part. Within the oxygenated blood, how much of hemoglobin is saturated with oxygen? That is what the point is. Now that 95. This is 10, 20, 30, 40, 50, 60, 70, 80, 90, 100. This line is 90, this is 100. 95, where it will come? For this 90 and 100, in between this, you will get the 95. When it is 95 mm mercury, what is the hemoglobin saturation with oxygen? 95, you just make a line here. It is almost it is coming to 100%. Yes or no? Yes or no? Observe carefully here. This is 90, 100, this is 95. Come up to this and you just come up to this. So what is the percentage saturation of hemoglobin with oxygen when it is 95? Though as per NCERT you are getting the answer as 100%, but actually it is around 98%. So here at this 95 mmHg, 98% of hemoglobin is saturated with oxygen. That is what the meaning is. From that particular graph, what you need? This is what the point you need. Clear? Next one. In the deoxygenated blood, what is the partial pressure of oxygen? It is 40 mm Hg. So when it is 40 mm Hg, just make the already line is there. You just come up to this. This is 60. This is 70. 70 is little bit above. So around 74, 75 you can consider. For convenience, I am taking the 75. Okay. Now let us see. At this particular one, what is the percentage of hemoglobin that is saturated? 75% hemoglobin is saturated with oxygen. That means in the oxygenated blood, what is the question here? In the oxygenated blood, see, 98% of hemoglobin is saturated with oxygen. But in the case of deoxygenated blood, 75% of hemoglobin is saturated with oxygen. This is what the meaning is. This is what the significance of the sigmoidal growth curve. So it's not growth curve, sigmoidal curve of oxygen dissociation. Understood or not? So in this way, this experimental oxygen dissociation curve we are using to understand these particular conclusions, because these particular partial pressures we are observing within the blood. Yes or no? Why in the experimental condition they are not going beyond 100? Because we don't want beyond 100, we want up to 95, that is enough. So that's why they made up to the 100, that's it. You don't need to increase further, there is no use where you will use this particular condition. We are not having more than 95, then what is the use in increasing that particular partial pressure of oxygen beyond 100? So you need to understand this. Why they have used that particular partial pressure up to 100 only? Because we need 95, that is maximum. After this we are not observing in our blood, then what is the use in making going further? But anyhow, at this 95 almost it is completely saturated. The hemoglobin that is present in our blood is almost completely saturated at this partial pressure. That's it. Even though you are further increasing partial pressure, there is no use. Yes or no? This is what the point is. So in this way, in the oxygenated blood, approximately entire hemoglobin is saturated with oxygen. In the deoxygenated blood, 75% hemoglobin is saturated. Understood or not? Further conclusions we will see in the next lecture. So now we will go further. This is what about oxygen dissociation curve. And please remember, this is the standard curve for adult one type of hemoglobin. This is the hemoglobin which is having alpha to beta 2 chains. But now if we are using this particular one in different conditions, first I will tell you in general, then I will tell you the specific example. This is the general example. What is the general example? Suppose let us take different molecules. Let us take different molecules. One is hemoglobin, one is hemoglobin one type, another one other type of hemoglobin, like with different type of molecules let us take. Suppose one molecule is showing like this and another molecule is showing like this. Suppose third type of molecule is showing like this. Let us take different, okay? Suppose it is showing like this. So now please remember, in all these cases the final value should remain same, even though here it is coming, for this somewhere it is coming and matching with the same, and here also it should come and match with the same. So all of them should be having the same level because total saturation will be same. So in this way, what we are saying here, in this way we are saying that this final value is 100% saturation. 100% saturation. Fine. For all the three, final saturation percentage is same only, 100% saturated. But for this, based on this, if you are talking about the 50% value, this is 50% saturated. 50% saturated. So 50% saturated value let us take. So on this graph paper, let us make the lines. Let us make the lines here. So on these lines, if you are talking, one line we are drawing here and it is cutting at this particular one. Let us make this as 1. And for the other one it is cutting at this location. Let us take this as number 2. For the third one it is cutting at here and let us give this number as 3. And what are these values? These are the partial pressure values. Partial pressure values. Partial pressure of oxygen. Yes or no? So here what is this partial pressure here? What are these particular values indicates? These will indicate P50 value. What is this P50? P is the partial pressure. This is the partial pressure value. P50 value for what? For 50% saturation. For 50% saturation, this is the 100% saturation. This is the 50% saturation. For 50% saturation, what is the partial pressure of oxygen required? For this first condition, this much is enough. Please remember, this is low partial pressure and right side is the high partial pressure. So I already told you, for all the three, 50% saturation is this one. But for this first condition, for the first type of molecule, to get 50% saturation, less partial pressure is enough. But for the second one, little bit more partial pressure is enough. For the third one, still more partial pressure is enough to get the same 50% saturation. That means This type of logical information I already told you when we studied about enzyme concept. Km values we studied, same logic. Their Km value, less Km value means more affinity to bind with substrate. Same logic here. Less P50 value means more binding affinity of oxygen to this molecule. That means in this case, this is the partial pressure of oxygen, right? At less partial pressure, at less value of oxygen itself, 50% saturated means oxygen has more binding affinity to bind with this molecule when compared with second one and when compared with third one. So if P50 value is less, oxygen has more binding affinity for this particular molecule. Understood or not? So if P50 value is more, means it has a less affinity to bind with this particular molecule. Same logic, same type of conclusions we studied for enzyme and substrate in the case of biomolecules chapter, and here we are studying for oxygen and the corresponding molecule. So in this way, P50 value is a significant value. So that's why P50 value will indicate what is the binding affinity of oxygen to that particular molecule. This is what the conclusion is. So in this way, P50 values are generally used. In this way, different P50 values will indicate that different affinities. That means in this case, for the blue line, whatever is the molecule is there. See here, blue line, high hemoglobin oxygen affinity. That means oxygen will quickly bind with this particular one. But if it is the red line, it is having very less. But the black one is intermediate. So in this way, please remember P50 values are very important in understanding the affinity of oxygen to bind with hemoglobin or any other molecule. This is what they are saying. And now these are the things that you need to understand. In this oxygenated blood 95 mmHg, so accordingly you can see the percentage saturation. Deoxygenated blood 40 mmHg, use this. At least these two things you should know. But for calculation purpose, other sentences that we can see in the next lecture. And here we are going with another example. Examples are important. Total 3 examples are there. What are these 3 examples? This is HbA. HbA means A1 alpha to beta 2 type. Okay, this is what. On the left side of this, you are writing about fetal hemoglobin. This is the HbF type. But here it is myoglobin. All the 3 I will explain. So very clearly, you please observe very clearly. Just now we studied P50 value. P50 value less means what? More binding affinity. Quickly it will bind. So just now I have explained in the beginning of the lecture about adult hemoglobin, fetal hemoglobin. Who has more binding affinity? Fetal hemoglobin has more binding affinity. Quickly it will bind. That means who will have less P50? Fetal hemoglobin will have less P50 when compared with adult hemoglobin. Now the same diagram I am drawing now. Observe carefully. Now let us draw the normal diagram for HbA. Specifically it is HbA1 having alpha to beta 2. Adult hemoglobin showing the normal graph. Fine. Okay. Now this is having a particular P50 value. P50 value, 50% saturation. Let us see this. Already here we studied, no? This one. From this only you can write this. This is for the normal hemoglobin. 100% saturation. This is 50% saturation. For the 50% saturation, you just come up to this. So what is this value? Approximately 27 mmHg, 27, 28 mmHg. Somewhere you are getting this 50 value. Just make it. Draw the line here. So you will get around 27 to 28 mmHg as per this particular given diagram. Yes or no? That is the P50 value for the normal one, normal hemoglobin. Yes or no? For 50% saturation it requires, suppose 28 mercury. This is the 50% saturation. This is the normal hemoglobin. But for fetal hemoglobin, on which side you will get? Will it exactly overlaps with this? Or is it coming on left side or going on right side? Please remember, fetal hemoglobin has more binding affinity. That's why it is coming on left side. So this will be same only. So this 50 will be same only. But when you are making a line, it is coming here less than this. Lesser P50 value. P50 value is lesser. What is the meaning of this? Fetal hemoglobin has more binding affinity. It will quickly bind with the hemoglobin when compared with adult hemoglobin. Yes or no? Oxygen will quickly bind with fetal hemoglobin when compared with adult hemoglobin. This is what the meaning is. This is what the significance of P50 value. And next, in the third example we are giving is myoglobin. For the myoglobin, you will not get sigmoidal. These two they are giving sigmoidal, but myoglobin you are getting like this. Why you are getting this? Why you are getting this hyperbolic curve like this? Because this is for myoglobin. Myoglobin, there is a important point here. Myoglobin has a single polypeptide, single protein in it. Myoglobin has single protein in it. But this hemoglobin, both the case it has four proteins. So here one myoglobin will bind with one oxygen. One myoglobin will bind with one oxygen. But here each hemoglobin will bind with four oxygen molecules. There is a difference here. So one molecule will bind with one oxygen, that's why quickly it is increasing and it is becoming like this. So that's why we are getting a different type of graph here. But in the case of hemoglobin, whether it is fetal or adult, whatever it is, now it is having two alpha chains and in adult two beta, but in the case of this one, two gamma chains are there. And here each one is binding with one, each one. So here tetramer we are talking, right? So each one is binding with one oxygen molecule. So four oxygen molecules we are getting here. And now you may think that why you are not getting straight line here? Why you are not getting straight line here? Why you are getting sigmoidal curve for hemoglobin here? Very, very important one. Even in the exercise also they have asked this question. And this here we are getting sigmoidal because of positive cooperativity. Because of positive cooperativity. Positively cooperative. What is the meaning of this positively cooperative? This positively cooperative means all the four are not binding at one time. So anyhow, one hemoglobin, the tetramer is binding with four oxygen molecules, that is correct. But first one will bind. For the attachment of the first one, attachment of first one is little bit slower. If first one is attached, now second one will not take that much time. For the first one attachment, suppose it is taking some x seconds time, suppose. For the second one it will take less than x. So it will be much faster. Once first one is attached and then second one is also attached. And when these two are attached, for the third one it will take further lesser time. When these three are attached, for the fourth one it will take still lesser time. So in this way, attachment of one oxygen molecule will enhance the binding ability of other oxygen to bind with the hemoglobin. So in this way, they are helping with each other. So that's why attachment of one will help in further attachment of the next one. So that is the reason why we are getting the sigmoidal curve. Please remember. But when you will get this straight line, here there is no positive cooperativity. For all of them, it will take equal time. For all of them, each one is taking the equal time. Then only you will get the straight line. But we are not getting straight line. We are getting sigmoidal. Sudden rise we are getting because there is a positive cooperativity. So attachment of one oxygen to hemoglobin will enhance or make it very quick to attach the next oxygen to the hemoglobin. So in this way we are getting the sigmoidal growth curve. These questions were given in the exercise also. You should compulsorily know the answer for each and every question here. So this is what this particular point is. Okay, so that's why it is giving the sigmoidal, because of the positive cooperativity. This is what the important concept that you need to understand. And with this particular information regarding the oxidation dissociation curve we are stopping. Further information regarding this we will see in the next lecture. So that in the next lecture we need to complete that transport of oxygen. Clear? With this lecture is completed. We will meet in the next lecture. Thank you.